We'll have a second talk, who is going to be delivered by Dr. Robert Malenka. Robert is the Pritzker Professor of Psychiatry, Director of the Nancy Pritzker Laboratory, and Deputy Director of the Wutai Neuroscience Institute at Stanford University. He graduated from Harvard College, where he received MD and a PhD degree from Stanford University. Then in the following 6 years, he completed psychiatry residency at Stanford and postdoc research at UCSF. So in 1989, he joined the faculty at UCSF and remained there until his return to Stanford University in 1999. He's the elected member of the U.S. National Academy of Sciences and the National Academy of Medicine, as well as the elected fellow of the American Academy of Arts and Science and the American Association for the Advancement of Science. He received numerous awards, including Kennedy Foundation International Prize in Neuroscience, the CIMP Lead in Neuroscience Basic Research Award, the Pearl UNC Neuroscience Prize, and the SFN Julie Axelrod Prize. So he has published hundreds of papers and been cited over 10,000 times. His work provides foundational knowledge about the mechanisms of synaptic plasticity and modulation in the mammalian brain. His laboratory continues to conduct research on the circuit mechanisms mediating adaptive and pathological motivated behaviors. So today he's going to give a talk entitled Serotonin and Sociability. Thank you. You can start. Yeah, you can see the slides.
I can't. Hold on. Can you hear me?
Oh, yes. Fantastic.
All right. Well, good morning, everybody in Beijing. And I don't know if there are people outside of Beijing. Thank you, Minmin, for that very nice introduction. I also want to thank Yirao and Xiang Yu, who have to take the credit and the blame for inviting me to give this talk. What I want to tell you about is work for my lab really over the last 10 years that has led us to pursue the hypothesis that serotonin release, in particular in a keynote of classic mesolimbic reward circuitry, the nucleus accumbens, plays a critical role in promoting what I'm going to term prosocial behaviors or sociability. And furthermore, I'm going to present evidence that decreases in serotonin release in this critical target structure play an important role in various types of sociability deficits in animal models of various brain disorders. So I became interested about 25 years ago in the role of classic mesocortical limbic reward circuitry, and in particular, VTA dopamine neurons and their projections to the nucleus accumbens in terms of their role in adaptive and pathological behaviors. I was particularly interested in elucidating whether various forms of synaptic and circuit plasticity play important roles in animal mouse models, primarily of neuropsychiatric disorders such as addiction and depression, because I was once a clinical psychiatrist who treated patients with these disorders. About 20 years ago, my lab began by administering drugs of abuse, such as shown here, cocaine, and looking for synaptic weight changes or synaptic plasticity in VTA dopamine neurons and nucleus accumbens. And I'd like to believe really about 20 years ago, we published a number of influential papers reporting that these synaptic plasticity mechanisms in key parts of mesolimbic dopamine reward circuitry do occur in response to drugs of abuse and play an important role in mediating the behavioral changes that accompany animal models of addiction. And I'd like to think that this led to a change in the way the addiction field thought about and studied neural mechanisms underlying addiction. So we continued to study various aspects of reward circuitry, elucidating in greater detail some of the heterogeneity in dopamine neuron subtypes, We looked at other types of synaptic changes in response to animal models of depression. But really beginning about 15 or 20 years ago, we started thinking about other experiences or stimuli in which the mesolimbic reward circuitry might play an important role. And one obvious stimuli or experience that may involve this circuitry are social interactions. For most of us, interactions, non-aggressive interactions with friends or colleagues and family are highly rewarding. It's also quite interesting that these same stimuli, for example, your wife or your partner, can be highly rewarding in 1 minute, and then 30 seconds later can be the most painful and aversive stimuli in your environment that you've ever experienced. And these changes in how social stimuli impact you do not happen magically. They must involve neural mechanisms that, I was really interested in elucidating. So the story, the work I'm going to tell you about, sorry, I'm trying to get my laser pointer to work. I'm going to tell you about really began now almost 15 years ago when a talented postdoc, Gul Dolan, joined my lab. And she came from a background of studying mouse models of autism. So she was really interested in some of the mechanisms that may contribute to sociability deficits in mouse models of autism. And we decided together to study the role of oxytocin action in the nucleus accumbens, which I'm really not going to talk about today, except to explain the hypothesis that was generated by her work and why that led to all of our interest in serotonin. So as shown in this slide, Gul, now almost 10 years ago, published a paper in Nature in which she used an assay for the reinforcing quality of a non-aggressive prosocial interaction in mice. And her data were all consistent with a fairly convoluted hypothesis that's outlined here. The idea she generated, or the hypothesis she generated, was that oxytocin release in the nucleus accumbens during a non-sexual, non-aggressive social interaction led to activation of oxytocin receptors on serotonin inputs in the nucleus accumbens. This in turn led to release of serotonin that activated specifically on serotonin 1B receptors that were found on excitatory inputs. So it's a pretty complicated hypothesis. All you need to remember from this is that the key downstream event or mechanism that we hypothesized was really important for regulating or promoting prosocial behaviors or sociability behaviors is the release of serotonin and its activation of serotonin 1B receptors. So that was the hypothesis we wanted to pursue. And there were the hypothesis suggested a number of what I would like to believe are fairly obvious experiments that were initiated by another talented postdoc who joined the lab probably 8 or 9 years ago and now has her own lab at University of North Carolina, Jessica Walsh. And what Jessica decided to do was manipulate serotonin neurons in the dorsal raphe nucleus. As all of you, I am sure, know, a major source of serotonin in the brain is the dorsal raphe nucleus. And specifically focus on the serotonergic inputs in the nucleus accumbens. And I will mention that all of the data I'm going to tell you about for the remainder of the talk was performed and analyzed blindly. And the reason I emphasize that is the behavioral effects Jess got and many of my other postdocs obtained that I'm going to tell you about are really quite robust. So the first set of experiments she did, and this work has been published now 5 years ago, so I'm going to go through it pretty quickly so I can get to some unpublished results, is she performed optogenetic manipulations of serotonin inputs in the nucleus accumbens. She expressed either channel rhodopsin or halorhodopsin, specifically in serotonin neurons, in the dorsal raphe by using a SERT-Cre driver line, in which Cre recombinase is only expressed in serotonin neurons. And then she ran the mice through a number of fairly simple assays of social behavior or sociability, a juvenile interaction assay in which you introduce a juvenile mouse of the same sex into the chamber with your experimental mouse, and you simply ask the experimental mouse, how much time do you want to hang out with the juvenile mouse in a non-aggressive manner? And then she also did the standard 3-chamber social preference or sociability assay, in which you take your subject mouse and you ask it whether it wants to spend more time in a chamber that has another mouse underneath a cup so they can't have any direct interactions, or does it prefer to hang out in a chamber that has an empty cup or a cup with an inanimate object. And then she also did a number of control behaviors that I won't have time to tell you about. And what she found is shown here. You're going to see a lot of graphs like this. The left graphs shown here show the individual animals, so you can see and decide for yourself and appreciate the variability and the reliability of the results. And the right-hand graphs show the mean and standard error of the mean of the results. And what this shows you is that in both assays, the juvenile interaction assay and the social preference or 3-chamber sociability assay, activation of serotonin inputs in the nucleus accumbens reliably and robustly promoted prosocial behaviors or sociability. Interestingly, inhibition of these serotonin inputs using activation of halorhodopsin in the nucleus accumbens serotonin inputs achieved the opposite effect. It caused a pretty reliable and robust decrease in these assays of sociability or social preference, which immediately raises the possibility that perhaps anything that reduces serotonin release in the nucleus accumbens might contribute to sociability deficits in pathological states. So that kind of thinking immediately led us to start wondering if such a deficit in serotonin signaling in the nucleus accumbens might contribute to the sociability deficits in autism spectrum disorder. So as you all know, there are many different mouse models of autism spectrum disorder. We decided we decided to study a mouse model of copy number variation known as the 16p11.2 deletion syndrome. This represents, according to my colleagues who study the genetics of autism spectrum disorder, anywhere from 1 to 3% of autism spectrum disorders, at least in the United States. But most importantly, we decided to study this mouse model because colleagues of ours at Stanford, Thomas Portman, who was working with Ricardo Dolmich, had generated a floxed mouse model of the 16p11 deletion syndrome. So that would allow us to genetically delete the syntenic chromosomal section in the mouse genome from specified cell types within the mouse brain. And that's what we did, and the results are shown in this slide. So just focus on these two graphs. This shows the decreases in juvenile interaction time or social preference when we took the floxed 16p11 deletion mouse, and we either injected a Cre virus into the dorsal raphe, so we're only deleting this chromosomal segment in the dorsal raphe neurons, or we crossed this floxed deletion mouse with the SERT-Cre driver line, so we're only deleting this chromosomal segment from serotonin neurons. And what's shown here is whether we injected the Cre virus in the dorsal raphe or we crossed the mouse with the SERT-Cre line, it caused pretty robust deficits in these sociability assays, suggesting that only deleting this chromosomal segment from serotonin neurons is sufficient to cause a sociability deficit. We then started looking at what changes. I'm sorry about this. My computer, for the first time ever, has just frozen. There we go. Whoops. I'm sorry about that. That's actually never happened to me. So So the first set of experiments we did was just to see whether serotonin neurons lacking this chromosomal segment show any physiological change. So we did some ex vivo slice physiology, and what this graph shows is that serotonin neurons in which this chromosomal segment has been deleted show a decrease in their intrinsic excitability. For a given current injection, they fire less spikes. What this shows you is simply that if we measure mini excitatory postsynaptic currents, on average, the amplitude of those currents in serotonin neurons lacking this chromosomal segment are smaller than control serotonin neurons. So there are some physiological changes that happen in these serotonin neurons that are consistent with some deficit in serotonin neuron activity. We then did this was our first try at doing fiber photometry. So in this experiment, we expressed GCAMP in serotonin neurons in either control SERT-Cre mice or SERT-Cre mice crossed to the FLOX16P. So these are mice that show the sociability deficits. And then during a social interaction with a juvenile, we measured bulk serotonin neuron activity using GCAMP. Time zero is when the mouse first approached the young juvenile and made almost physical contact. And what you can see here is that in control mice, there's a fairly clear increase in serotonin neuron activity during a non-aggressive social interaction, and that on average, that is reduced in this mouse model of autism spectrum disorder. So again, that's consistent with the previous results I just showed you, showing decreased excitability of the serotonin neurons, decreased strength of excitatory input. So we then wondered if it is the case that a deficit in serotonin neuron activity during a social interaction, and presumably a deficit in serotonin release in the nucleus accumbens, contributed to the sociability deficits in this autism mouse model. Could we rescue the sociability deficits by activating serotonin inputs in the nucleus accumbens? And of course, I wouldn't be telling you this if it didn't work. So in these graphs, all of the mice are the autism mice. All of these mice are the SERT-Cre mice crossed with the FLOX16P mice. So you can see from the control mice in which we expressed Y of P in the serotonin neurons, all the mice show a pretty profound sociability deficit. But when we shine the light and presumably in the nucleus accumbens and presumably activate serotonin neurons, serotonin inputs in the accumbens, we get a very robust and very reliable rescue of these sociability deficits in both the juvenile interaction task and the 3-chamber social preference task. Now, remember, I told you that the original hypothesis that Gould-Dolan's work suggested that serotonin was working in the nucleus accumbens, at least in part, by activating serotonin 1B receptors. So we wanted to know whether this rescue of the sociability deficits in these autism mouse model was due, at least required activation of 1B receptors in the accumbens. So the experiment just did is shown here. She now expressed channel rhodopsin in serotonin neurons in these autism mutant mice. She shined the light in the dorsal raphe, so she's activating the cell bodies, and that allowed her to infuse either saline or a serotonin 1B receptor antagonist into the nucleus accumbens. And what this shows you, this is a different cohort of mice. So again, all of these mice have sociability deficits. When she activates the serotonin neurons and infuses saline, the black lines here, she gets a beautiful rescue of their sociability deficits. But when she infuses the serotonin 1B antagonist, she completely blocks the rescue of sociability deficits. So that then raised the question of whether we could use a serotonin 1B receptor agonist infused into the nucleus accumbens as a pharmacological way of rescuing the sociability deficits. And that's what's shown here. So again, these are the green lines show the autism mice, the 16P deletion mice. that again show this robust sociability deficits. And you can see that infusion of a 1B agonist again rescued their sociability deficits. So finally, in more recent work, we wondered about the clinical relevance of this. So we obtained a serotonin 1B agonist that we could administer IP into the bellies of the mice. And in a recent publication, we reported that this manipulation rescued the sociability deficits. It was actually in 6 different mouse models of autism. In this case, I'm just showing you 3 different mouse models. The details actually aren't important. It's a contactin knockout mouse, the Fragile X, a model of Fragile X syndrome mice, and an environmental model of autism deficits in sociability. And I should say, based on these kinds of findings and many others from my lab, a biotech which I work with is about to initiate phase 2 trials testing the efficacy of a serotonin 1B agonist in human beings with autism spectrum disorder. Okay, so what I've shown you so far is that bidirectional modulation of serotonin release in the accumbens bidirectionally influences prosocial behaviors. Deletion of this chromosomal segment from serotonin neurons decreases their prosocial behaviors, decreases their excitability, and that we can rescue these sociability deficits by causing release of serotonin in the nucleus accumbens. Okay, around the same time Jess was doing this work, another postdoc joined my lab, Boris Heifetz, who's an MD, PhD anesthesiologist. And it turned out we had a shared interest in pursuing the mechanisms of action of a drug that also is well known to promote prosocial interactions in human beings. And in fact, some people claim that it actually promotes empathy in human beings. And that's a drug known as MDMA, or by its street name in the United States, ecstasy or Molly. I don't think I'm not sure it's popular in China, but in the United States, millions of individuals have taken this drug recreationally. And we were interested in it for several reasons. We were interested in pursuing its mechanisms for several reasons. First, it's currently being evaluated as a treatment, as an adjunct to psychotherapy for post-traumatic stress disorder. It's also being tested in individuals with autism spectrum disorder. So there's a lot, and the results look very promising. So that was a motivation for trying to understand its mechanism. But thinking mechanistically, we were particularly interested because its main actions that have been reported are, it's well known to cause a dramatic and robust release of serotonin. So it's a pharmacological way of doing an optogenetic experiment of causing the release of serotonin. It's been suggested to influence oxytocin levels, and it also can cause a more modest release of dopamine. So Boris decided to start studying, and the first thing he needed to do was make sure he could develop an assay in which MDMA enhances social exploration or sociability, whatever words we want to use. So again, he turned to the 3-chamber sociability or social preference assay. What this shows you in the upper right here is a dose response of the effect of giving the mouse MDMA. And you can see at 7.5 milligrams per kilogram or 15 milligrams per kilogram, it caused a robust enhancement of social preference. This bottom right panel shows what I think is a cute experiment. In this experiment, in this bar graph, he only gave MDMA to the mouse that was under the cup, and he got a modest effect. If he gave MDMA to the subject mouse that was allowed to explore, he got a pretty clear and bigger effect. And most interestingly, if he gave both mice MDMA, he got the most robust effect. And this actually very much mimics the experience of human beings taking this drug. So how does MDMA work? Well, we were also, before telling you that, we were also interested in the abuse liability of MDMA. MDMA is a psychostimulant. And as I mentioned, it can cause the release of dopamine, which means it has abuse or addictive liability. So we wanted to see if we could find doses of MDMA that were prosocial but not addictive. or at least wouldn't have as much addictive liability. And the simple way to do that was to do two different assays. The first assay was just a locomotion assay. It turns out that every single drug of abuse that has addictive liability in human beings and addictive liability in mice also increases locomotion. And it turns out that at 7.5 milligrams per kilogram, MDMA doesn't increase locomotion, but if you'll remember, it did enhance social preference in the 3-chamber task. But at 15 milligrams per kilogram, MDMA causes a robust increase in locomotion. Perhaps more importantly, we did a conditioned place preference assay, which is a standard assay for testing the reinforcing quality of a drug experience. And again, consistent with the motor effects, a dose of 7.5 mg per kg did not cause conditioned place preference, but a dose of 15 milligrams per kilogram did. So we could use these two doses to try to dissociate the abuse liability of MDMA from its prosocial effects. Okay, now you remember that I told you that MDMA causes a really robust enhancement of serotonin release. That's been known for a long time. And it does this because it binds and interacts in a way that's very different than SSRIs like Prozac, but it interacts with the serotonin transporter. It actually causes reverse transport of serotonin and forces serotonin, excuse me, out of serotonin nerve terminals. So if MDMA interacting with the serotonin transporter was required for its prosocial effects, preventing that interaction should prevent MDMA's prosocial effects. So we did that experiment two different ways. We first did a simple pharmacologic experiment where we gave the animals a drug known as citalopram that is known to bind to the serotonin transporter in a very different way than MDMA, but it prevents the binding of MDMA to the serotonin transporter. And you can see that blocked the enhancement of sociability by MDMA in the 3-chamber task. We then were able to attain a conditional knockout mouse of the serotonin transporter from Dr. Zhe at Albert Einstein Medical School. So then we could knock out the serotonin transporter only from dorsal raphe neurons. The way Boris did that is he actually injected a Cree virus into the dorsal raphe. And when he did that, what this shows is if he knocks out the serotonin transporter only in dorsal raphe neurons, he can pretty much, on average, block the enhancement of sociability by MDMA. But importantly, the knockout of the serotonin transporter from dorsal raphe neurons did not block the conditioned place preference elicited by a higher dose of MDMA, suggesting that the interaction of MDMA with the serotonin transporter is not important for its rewarding component at higher doses. And we actually now know that's due to interactions with the dopamine transporter. So those simple experiments suggested that it was MDMA interacting with the serotonin transporter, presumably causing an enhancement of serotonin release that was required for its prosocial effects in this 3-chamber assay. So to test whether it was MDMA's action specifically within the nucleus accumbens, Boris did, again, what I hope is an obvious experiment to all of you who are neuroscientists in the audience. That is, he infused MDMA directly into the nucleus accumbens. And what this shows is, here's a group of animals that received MDMA IP, a reasonably robust effect. Excuse me. Here's what happens when he infused MDMA directly into the nucleus accumbens. He saw a very robust enhancement of social preference in the 3-chamber assay. Conversely, he could do the complementary experiment. So what he's doing here is giving MDMA IP into the belly of the mice and either infusing saline or citalopram, this drug that binds to the serotonin transporter and prevents MDMA from binding to it. And you can see that infusing citalopram only into the nucleus accumbens on average block the effects of MDMA. So together, we think this is reasonably strong evidence that just like the optogenetic activation of dorsal raphe serotonin neurons and the way it's causing an enhancement of serotonin release in the nucleus accumbens, MDMA is doing the same thing, but doing it pharmacologically. And then finally, we were interested in whether this is the evidence I was talking about previously, whether MDMA's abuse liability, that is, its reinforcing characteristic at higher doses, had anything to do with the serotonin transporter or dopamine action in the nucleus accumbens. So what this data shows you is that infusion of citalopram into the nucleus accumbens did not block the conditioned place preference generated by administration of MDMA, but infusion of a dopamine receptor antagonist into the nucleus accumbens on average blocked the conditioned place preference caused by MDMA. So together, these data support the kinds of conclusions I talked about previously. So again, because we're interested in the translational implications of this kind of work, we were also interested in whether, like a serotonin 1B agonist, whether MDMA itself would influence sociability deficits in mouse models of autism spectrum disorder. And so what Jess Walsh did is take some of the same mice which she had studied before and saw an even more robust rescue of sociability deficits in 3 different mouse models of autism spectrum disorder. And I should say, based on these kinds of results, there's a different drug company called MindMed who is pursuing a specific enantiomer of MDMA and is hoping to test it in human subjects at some point in the next 2 years, subjects with autism spectrum disorder. So I'd like to believe this kind of translational work has stimulated testing new types of therapeutic approaches to the treatment of sociability deficits. Not only in autism spectrum disorder, but perhaps in other brain disorders that involve sociability deficits. So I hope I've answered this question that I introduced the talk with, which is, does serotonin and the release in the nucleus accumbens promote prosocial behavior? And we think the answer is a resounding yes. So at this point, a few years ago, I really wanted to see, because remember, I originally became interested in this mesolimbic reward circuitry because of its potential and plasticity in this circuitry, because of its involvement in brain disorders such as addiction or depression. And I wanted to see if we could connect this type of work on sociability with one of those other disorders. And I was fortunate to have another postdoc join the lab about 3 years ago, Matt Pomerantz, who came from an addiction background. He had been studying mouse models of addiction during his PhD, in particular opioid addiction. What's shown here is a simplified model of some of the steps that may occur in human beings with addiction. They initially start binging and taking their chosen substance, whether it's alcohol or heroin or fentanyl or cocaine or methamphetamine. They binge, they become dependent. At some point, they stop using it and they try to become abstinent. And what many of you may know about the challenges of treating addiction is the major challenge is not getting addicts to stop using their drug. It is once they have stopped using their drug, even if it's for weeks or months, it's to prevent them from relapsing. It's to prevent them from starting to use the drug again. And one hypothesis about this prolonged abstinent state has been proposed by George Koob, who's the director of the Alcohol Institute at NIH in the United States. And he's proposed that addicts go through with an emotional withdrawal state that has many similarities to depression. Many of the symptoms that addicts experience overlap with depression. And he's called this state, for reasons I don't have time to explain to you, hyperkataphia. That's not important. What is important is that the constellation of symptoms addicts experience include anhedonia, anxiety, dysphoria, and very of interest to us is strong social avoidance and isolation. What's also of interest to us, I don't know if you're going to be able to see this, is George Koob and the director of the National Institute on Drug Abuse here in the United States, Nora Volkoff, have also proposed some of the circuit changes that occur during this protracted withdrawal from drugs. And a key component of that circuit pathology is the ventral striatum or the nucleus accumbens. So my new postdoc, Matt Pomerantz, knew all this. He knew I was interested in the role of serotonin and sociability. He wanted to work on something related to addiction. So he decided to develop a model of protracted drug withdrawal, in this case, protracted withdrawal from dependence on morphine. And here's the protocol he did. And again, he was specifically interested in the sociability deficits elicited during this protracted opioid withdrawal. So he tested the animals in these standard assays, the juvenile interaction 3-chamber assay. He gave the animals escalating doses of morphine, which is a standard protocol to get the animals dependent and allow them to go through a severe withdrawal state. He then withdrew the animals from morphine for 3 weeks and tested them. And you should be used to looking at these sorts of graphs by now. And what you can see is that both in the juvenile interaction task and the 3-chamber social preference task, animals going through this protracted, prolonged opiate withdrawal state show very robust sociability deficits. Okay, so how do we attack mechanism here? I don't have time to give you all the reasons we decided to start focusing on the role of kappa opioid receptors in this sociability deficit in protracted opioid withdrawal. One reason Matt was particularly interested in kappa opioid receptors was there's already an extensive literature suggesting that kappa receptors in VTA dopamine, in the VTA, in the nucleus accumbens, in these key nodes of reward circuitry, that kappa receptors play an important role in triggering aversive states and also play an important role in adaptations to chronic stress, which many of you know is a model for eliciting depressive-like symptoms. He was also interested in kappa receptors because kappa opioid receptor antagonists have been suggested by NIDA to be worthwhile exploring as treatments for addiction. And furthermore, kappa receptor antagonists are currently being tested in human subjects, in this case, as a treatment for one of the key symptoms of anhedonia. So he hypothesized that this protracted opioid withdrawal state involved kappa receptors in the nucleus accumbens, and that they may contribute to the sociability deficits that he had seen. So I only have a few minutes left. I'm going to rush through this. So the first experiment he did is he infused a kappa receptor antagonist into the nucleus accumbens and asked whether that influenced the sociability deficits during protracted opioid withdrawal. Because this antagonist is irreversible, he had to do it in a manner where he could control for that. So what he did is he first made all the animals opioid dependent. He withdrew all the animals from the morphine, and you can see all the animals showed these sociability deficits. Then half the animals received a saline injection to the nucleus accumbens, and the other half of the animals received this kappa receptor antagonist. And you can see the saline-infused animals still show the sociability deficits, but the animals that received the kappa receptor antagonist into the accumbens, the sociability deficits were completely reversed. Many of you probably know that the main endogenous ligand for kappa receptors is the opioid peptide dynorphin. He was interested in where the dynorphin derived from that might be responsible for activating these kappa receptors during opioid withdrawal. An obvious source of those were the medium spiny neurons and accumbens themselves. It turns out that blocking dynorphin release using tetanus toxin from medium spiny neurons did not prevent the sociability deficits. So he then searched the brain for cells that express dynorphin and found that, surprisingly, there was a population of neurons in the dorsal raphe that expressed dynorphin. He then used a dynorphin Cree mouse and injected a Cree-dependent tetanus toxin so he could block release from these dynorphin neurons. And what this shows is that expressing tetanus toxin in dorsal raphe dynorphin neurons completely prevented the sociability deficits during protracted opioid withdrawal. That suggested that perhaps dynorphin release itself in the nucleus accumbens might cause a sociability deficit. I hope the logic is clear there. I'm speeding up because I don't want to go over. So he did the experiment in which he expressed channelrhodopsin in the dorsal raphe dynorphin neurons using this dynorphin Cree mouse. And what this shows is that when he activated dorsal raphe dynorphin inputs in the nucleus accumbens, he caused a robust sociability deficit. So that then raised the question of where are the kappa receptors that are presumably being activated by the released dynorphin? Where are they localized? And again, because I'm going over, I'm not going to explain why we did all these experiments. The key experiment is the one shown here. We hypothesized, it was known that there are kappa receptors on serotonin neurons, and it had been previously reported that kappa receptor activation can cause a decrease in serotonin release from serotonin inputs in target structures. So we hypothesized that maybe what's happening during protracted opioid withdrawal is that dynorphin levels are increasing in the nucleus accumbens. They're activating kappa receptors, specifically on serotonin inputs, causing a decrease in serotonin release. And that should, according to all of our previous work, cause sociability deficits. So if that's the case, knocking out kappa receptors from serotonin neurons should prevent the sociability deficits during opiate withdrawal. And that's the results he got here. So what's shown here is control mice showing the normal sociability deficits during opioid withdrawal, and then mice in which he has crossed a Cert-Cree mouse with a mouse that has the kappa receptor flock. So it's a conditional knockout of the kappa receptor. So he's deleting the kappa receptors only from serotonin neurons. And you can see those mice don't cause sociability deficits. So to wrap up, we wanted to more directly test our hypothesis. And these are my last 2 or 3 slides. I want to have a shout out to Yulong Li. I don't know if he's listening here. As many of you know, he's a Beijing neuroscientist who has really advanced our ability to directly measure neuromodulators by developing these genetically encoded sensors of neuromodulators called GRAB dopamine, GRAB serotonin, GRAB acetylcholine. So we used his GRAB serotonin. We expressed it in the nucleus accumbens and then used fiber photometry to measure serotonin release by the fluorescence of GRAB serotonin. What this shows is a confirmation of what I mentioned earlier. Remember, I said a major effect of MDMA is to cause a massive release of serotonin. And what this shows is if we actually measure it more directly using GRAB serotonin in the nucleus accumbens, it does indeed cause a very prolonged massive increase. So the GRAB serotonin does work. So what happens during a social interaction? Now, remember, I also showed you previously that during a non-aggressive social interaction, we see an increase in the activity of serotonin neurons when we measure it using GCAMP. This is a complementary experiment. Now we're measuring serotonin release in the nucleus accumbens directly. what this shows is that during baseline testing, we see an increase in serotonin. And these are all the animals before any of them have gotten morphine. If we give one group of animals the morphine, let them go through withdrawal and compare them to the saline animals, you can see that during the second sociability testing, that the control animals still show an increase in serotonin in the nucleus accumbens, but on average, the animals that are going through opioid withdrawal that show sociability deficits don't show that increase in serotonin. Now, if this lack of serotonin increase during the social interaction is due to activation of kappa receptors on serotonin neurons, I know that's a lot, we should be able to prevent this change in the serotonin release in the nucleus accumbens by knocking out kappa receptors in serotonin neurons. So that's the experiment we did. So what Matt did is he again crossed the CERT-CRE mouse with a conditional knockout of the kappa receptor. He then expressed grab serotonin in the nucleus accumbens. This is a different cohort of mice. This shows their baseline serotonin release increases in the nucleus accumbens. And I should mention, and then all of these mice went through the morphine procedure. So all of them in this second measurement are now going through withdrawal. And what this shows is that the control mice showed the same lack of serotonin increase during the protracted opioid withdrawal, but the mice in which we knocked out the kappa receptors from serotonin neurons show a roughly normal increase in serotonin in the nucleus accumbens. I'm going to skip that. So the summary is that the sociability deficits during protracted opioid withdrawal requires dynorphin release in the accumbens from this population of dorsal raphe dynorphin neurons, which raises some hopefully obvious questions. That we think, this is a hypothesis actually, that the dynorphin activates kappa receptors on these dorsal raphe serotonin inputs to decrease the serotonin release. And what this says is that we have two different, if we combine the previous results I showed you, is that we can see sociability deficits in an autism mouse model as well as during protracted opioid withdrawal. And they both seem to involve decreases in serotonin release in the accumbens, but it's happening via different mechanisms. So with that, I just want to acknowledge all the people who did the work. I think I mentioned them as I went along, and I'm happy to answer any questions.
Thank you, Rob, for a great question. Great talk. There are already some questions from the audience. Actually, I just want to let you know the talk was broadcast live in several platforms. And we received several questions. So let me compile them. And one of the key questions from the audience is that many of them are very interested in MDMA, knowing that it can increase sociability. So the questions are like whether the effect is sustainable, whether it has any side effects, and whether since it's target, serotonin, increased serotonin, whether the standard SSRIs will have similar effects.
Okay, that's a good question. So the reason we think SSRIs have very different effects has to do with how they molecularly interact with the serotonin transporter. And as a consequence of very different molecular interactions, the effects they actually have on the release of serotonin, the buildups of serotonin in target structures. So, and there's a lot of evidence to support what I'm going to say. So SSRIs like Prozac only inhibit the serotonin transporter. They don't cause reverse transport. They don't do anything like that. So what's going on is they're only preventing serotonin that is being released by action potential dependent release from being taken up. So if you imagine, let's say, a sink, it's sort of, and you put a stopper in the sink and the sink is dripping very slowly, you get a very slow buildup of serotonin. that is taking probably hours or days. And that very well may be why it takes a week or 2 weeks or 3 weeks for SSRIs to work. And in fact, if we measure GRAB serotonin when we give an animal an SSRI, we basically almost don't see very much. We see something happening, but nothing like MDMA. MDMA not only blocks the serotonin transporter, as I said, it causes a reverse transport, kind of a massive release. So the kinetics of serotonin increases in target structures are orders of magnitude faster and larger. And it's a good graduate student question. Why would a very rapid, large increase in serotonin, you can imagine, have very different behavioral actions than a very, very slow buildup of serotonin? And this kind of thinking is exactly the reason why we think drugs of abuse like amphetamine or cocaine or opioids are highly addicting, but many other rewarding experiences aren't nearly as addicting. And this is based on Nora Volkoff's work, that the kinetics of dopamine release in the accumbens are much larger and much more rapid with psychostimulants or opioids than when, for example, you eat an ice cream cone or you have sex. So I hope that answers the question. Now I forget the other parts of the question.
Oh, whether it's sustainable and whether the benefit is sustainable and whether it has any side effect.
Yeah. Well, with MDMA, as I said, because I think it will get approved for PTSD in the next year or two in the United States. We are concerned about its abuse liability. I don't think it will be as addictive as cocaine or amphetamine, but I do think with inappropriate use, it can cause problems. It can also cause toxicity. And that's why we're really interested in elucidating the mechanisms, because by understanding the mechanisms, for lack of a better way of saying it, can we make a better MDMA? So the question is, are the effects long lasting? Unfortunately, we don't think so. So we did a lot of work, because some of you may know, I used to study plasticity. I used to study LTP and LTD. I was hoping a single injection of MDMA would cause effects that outlasted the drug experience itself. But we never saw that. We did a lot of studies where we gave MDMA to our normal mice, our autistic mice. We tested their social behaviors while they're presumably experiencing the MDMA. Then 8 hours later, we tested them a day later, and unfortunately, we never saw carryover effects. And that's why I think, to be honest, that's why my drug company that I advise, I should say, is pursuing a 1B agonist rather than MDMA, because we know we can give that daily and it seems pretty safe. And that's why another drug company I happen to advise, called MindMed, is pursuing a certain form of MDMA that we don't think has nearly as much abuse liability. So we're hoping it can be given more frequently. So I hope I answered the question. But I also want to make it clear, that doesn't mean MDMA doesn't have any long-lasting effects. It just means we weren't able to detect them in the assays we performed.
So due to the time limit, we'll reserve the last question for Dr. Xiang Yu. She asked whether you have seen any difference in optogenetic stimulation of nucleus accumbens terminals, or between that optogenetic stimulation and drugs application of MDMA. Or
that's a good question. So differences between optogenetic activation of the inputs and MDMA infusion. We haven't, but we have not done a thorough exploration of that. I mean, to be honest, we were just thrilled. Obviously I'm reviewing, this is 8 years of work, actually. It was reassuring to us that we saw similar effects with our very admittedly primitive, simple assays of sociability. So there very well may be some differences, but we haven't pursued those.
Okay, great. I think, Rob, before you leave, I want to let you know that right now we have 23,000 audience for your talk.
No. How many?
Yeah,
2,000? Did you say 2,000 or?
23,000.
Congratulations. And again, thank you for having me. I appreciate it. And sorry I went over.
No, not a problem. Thank you so much for a great talk. I look forward to having you visit us when it allows.
Well, if we can get our governments to cooperate, that would be. Am I allowed to say that? Whoops, I probably shouldn't say that.
No,
I'm just
great.
I'm just a lowly scientist.
You'll be okay.
All right. Anyhow, thank you. I'm going to stay and listen to Nancy.